There we are, we're recording now. So hello everyone and welcome to the next episode of My Science Podcast. Today I am delighted to welcome Professor Florian Engert, who is the Professor of Molecular and Cellular Biology at Harvard University. His research entails the neural circuits and behaviour in larval zebrafish. His lab is working on a scientific strategy focused on building a complete multi-level picture of simple neural circuits that will advance our basic understanding of brain function and offer a complete view into the neuronal activity underlying a series of relatively complex behaviours. And so thank you very much for joining me today.
It's a genuine pleasure.
So to start off with, may you give us an outline of your career and why you entered biology as a profession?
Yes, so I started my career in physics and philosophy during college, and I think that was driven by a drive to get a basic understanding of the world as such, and I thought that physics and philosophy would give an answer to that. And then during the final stages, the diploma part, I realized that the actual research in physics, so philosophy I dropped relatively quickly, it was just too much, but then in physics the two fields that were available at the time were particle physics and astrophysics. And that seemed quite remote from daily life. It's either too small or too big. And there was an opportunity to branch out into neurophysiology because they were also looking for physicists, and I think mostly for technical reasons, right? They needed people who are familiar with the technology. And neuroscience, I think, was fascinating because it is close and immediate to your personal existence almost, right? I think thinking about how the brain works is immediately relevant and intimate because we all have brains. And so I think that seemed like a very close aspect. So I did my PhD then in neurobiology in Munich. And then initially I studied single nerve cells, and then I slowly climbed the ladder of complexity. During my PhD was then brain slices. My postdoc in California was done in tadpoles, xenopus tadpoles. And then for my first junior faculty position, I moved from tadpoles to zebrafish, just because it became, it sort of seemed more relevant to study intact brains rather than individual neurons.
So coming off what you said, in your research, why do you use zebrafish rather than any other organism?
Yeah, so I think the power of the zebrafish, and it's babies, it's larvae, it's not the adults, right? We study zebrafish larvae, and the main power is it's tiny. They are not more than 1 millimeter across and 4 millimeters in length. So they fit comfortably under a standard light microscope. So you can look at the whole animal in its entirety. But more importantly, they're translucent. So you can look right into the animal. You can look into the brain, which I think that's the biggest advantage. You can see. You don't need to open up the head in order to see the brain. It's the lack of opacity is very powerful. And like many other animals, it's a transgenic model system. So you can use genetic tools to make the neurons fluoresce on their own. That's not exclusive to zebrafish, but in combination with the small size and transparency, the ability to introduce genetically programmed reporters, fluorescent reporters, where you can read out activity just by fluorescence. So in combination with the translucence and the small size, I think makes the zebrafish unique. Yeah. And the other thing that makes it attractive is that they do have a relatively complex repertoire of behaviors. So they're using their brains for useful, for applicable reasons. And lastly, they're vertebrates. So other than worms or insects, they are card-carrying vertebrates. So the brain structure of a larval zebrafish, even if it's tiny, is very, very similar to that of a mammal or even a human.
Absolutely. So what technology allows you to monitor neural activity throughout the fish brain in an awake and intact preparation?
I think the simplest aspect of technology is just microscopy. Just simple light microscopy will allow you to do that. That's not quite true. It is the fluorescent microscopy. So standard light microscopy, you just use normal light to visualize your sample. In fluorescence microscopy, you use two different wavelengths for excitation and emission, and that enhances by a huge degree the contrast and the resolution. So that's what we use. We use fluorescence microscopy, which is a very old technique. And there are more fancy and elaborate aspects of fluorescence microscopy. One of them is confocal microscopy, and a more modern one is two-photon microscopy. Yeah. And that's what we're using. Yes,
how do you go about relating an input stimuli, for example, visual signals being translated then into actional potentials by the retina and ganglion cells, to the output of a motor action controlled by spikes in motor neurons?
Well, I think that is the fundamental question. You're right. I think this is where the rubber hits the road. I think this is the computation that all of our brains are performing. I think any brain, right, does exactly that. It takes an input and patterns of inputs and translates them into the adaptive and adequate patterns of outputs. And this is done by neural circuits. Right? This is done by in the same way that electronic circuits do certain computations, maybe even in your iPhone and in your computer. The brain is doing something similar. And I think that this is the name of the game. Yes. How do these biological neural circuits perform these computations that translate one set of spikes, like the ones represented by the input? How do they get transposed and translated into an equivalent set of action potentials that generate the adaptive output and the behavior?
So you've touched on this a little bit already, but what are genetically encoded indicators, and how has this technique allowed you to go about recording activity in every neuron in the fish's brain in the context of a specific behavior?
Yeah, so this was a fundamental breakthrough in biology, the discovery of these indicators. And originally they've been isolated from jellyfish. These are the green fluorescent proteins that you can see in the ocean at night when there is fluorescence in the ocean. This is usually done by microorganisms, jellyfish-like organisms. We still don't know why they glow, but they do. Yes, so they can generate these proteins that glow in the dark. And I think what has been done maybe 15 or 20 years ago by Shimomura and Martin Chalfie and Roger Chen, they collectively got the Nobel Prize for first extracting these proteins from jellyfish, then sequencing the genome, knowing how to how it's encoded by the genome, and then inserting these genomes into any other organism. For the first time, it was done in C. elegans in a flatworm. But once you know the code, the genetic code that encodes for these green fluorescent proteins, you can put them into any organisms. And then you can even ask these organisms to generate these green fluorescent proteins only in the cells that you're interested in, such that not the whole animal glows, but just the neurons. So this was the fundamental breakthrough, yes, that we asked the animal to make these green fluorescent glowing proteins. And then I think a secondary advance and breakthrough was to have these proteins not glow all the time, but only when the neuron is active. Yeah. So this is sort of the great idea, is to couple the emission, the light, the fluorescence of these proteins to the activity of the neurons. And the way this was done is by using calcium, the element calcium, as an indicator, as a second messenger. So calcium is something that goes up in an active neuron, and only when the neuron is active.
Yeah.
And there are other ways of coupling this influx of calcium to the fluorescence of the indicator. And that's how you can make them activity dependent. That was the second trick, right? First, make them glow. And then the second step is to make them glow in an activity dependent manner.
That's absolutely fascinating. So may you explain what the optic tectum and the reticular formation are and how these link to your research?
Yes. So all vertebrate animals, including mammals, including us humans, have a structure in the brain that is responsible to process part of the visual information. Yes. So all of these animals, including us, have several brain areas that are responsible for processing separate aspects of visual information. The superior colliculus, in our case, in vertebrates, it's also called the optic tectum, is one of these brain areas that is responsible for receiving visual information from the eye, processing it, extracting information, and then sending it onwards. And so what in humans is called the superior colliculus, in lower vertebrates is called the optic tectum, and that is what's processing sort of fundamental aspects of visual information. Most animals use this information, send it onward. In the zebrafish, it's called the reticular formation, where motor programs are being generated.
So my next question is, how do zebrafish use the olfactory system to avoid high salinity as they lack epithelial sodium channels?
Yes. Oh, but that's, yes. They lack epithelial sodium channels. So this is back in my area of expertise. This is something we did. So detecting salt is something that we all do through taste, and it's important because we need salt in order to survive. So mammals have evolved an affinity to salt, but we detect salt with our taste organ, with the tongue. We found recently that zebrafish smell salt. They don't use that. They can also detect it with the tongue. They also eat, so they need to check for salinity in the food they consume. But for a fish, detecting salt is also important, independent of what you consume, because there are freshwater fish and saltwater fish, and if a freshwater fish gets into regions of high salinity, that's really bad, right? They die. So they need to detect salt in the water to survive instantly. And so it wasn't known how they do that. And it's not really epithelial. So some of the salt detectors on the tongue are epithelial salt receptors, but zebrafish can also, in principle, detect salt through the skin or the lateral line, the hair cells. But we found they can actually use the nose to detect salt, so they can smell it. And that seems to be a very powerful way for them to quickly detect regions of high salinity or gradients of high salinity and avoid them.
Absolutely. So your discovery of the homeostatic control of that larval zebrafish use phototaxis to maintain environmental luminescence at a set point that depends on luminescence history has given rise to a behavioural algorithm by which they exert control over this sensory variable. So what further research may come of this?
Yes, this was a project that had, where we are asking almost two separate questions. One is how do fish, larval zebrafish, detect areas that are of higher brightness and swim there? So fish were thought to be simple phototactic animals, meaning they like the light and avoid the darkness, like moths and flies at night. And the reason why they do that is relatively straightforward, because they're very visual animals, so they rely on vision. So they swim where it's brighter, so they can see, they can hunt, they can escape. So this was a further study into how fish detect the regions in the environment that are brighter, and how do they navigate to swim there to maximize brightness. This was our original hypothesis, that they do that, and we find they do, and we made some progress in uncovering what precise algorithm they use. The second part, so this story became more complicated, because what we realized is that fish don't just swim to wherever it's brighter, but they have a preferred overall brightness level that they like to hang out. So if it's getting too bright, they also don't want that. And this is where the homeostatic regulation came in. So they prefer a set point of brightness that is ideal for them, and so they can change the preference for whether they want to swim towards brighter regions or darker regions, depending on the history. This is a little bit like a thermostat in a building, right? That we have a set point is the temperature that you would like to achieve. If it gets too hot, you need to cool. If it's too cold, you need to heat it. So this is a dynamical system, a feedback system that is under homeostatic control. This falls under the whole field of control theory, which is fascinating. And so what we found is that fish also have this control system, this dynamic control system for an overall level of brightness that they prefer. Yes, and so therefore this was almost a twofold project, right? One is this immediate way of swimming where it's brighter, but then under a larger picture, it was about how they do homeostatic control, and also when it comes to overall brightness and luminosity.
That's amazing. So these fish have 4 types of cone photoreceptors, covering most of the visible range and into the UV. And so what specific combinations of response types are localized to specific nuclei when stimulating the fish with different colors?
Yeah. So that's interesting, right? Fish can actually see color better than us. And they have an additional UV cone, meaning the 4 cones are the 4 different receptors with which they can detect different colors. So like humans have red, green, and blue cones, channels, and color channels, and fish have an extra UV one. And I think that the main idea here is that fish, because they have evolved in Pakistan and India, are subject to intense UV radiation. They get sunburned very easily. As I've told you earlier, they're also translucent, so they are extremely susceptible to damage by UV radiation, and that's why we believe have they evolved an independent detector for UV, such that they can avoid this with higher sensitivity. So that's the underlying idea, that the UV channel in the fish's eye is like a danger signal, a specific targeted danger detection channel. And that's something we tried to elucidate in more detail.
Right. And you've also done studies on around the fish's appetite. So when you explored the medial and lateral hypothalamic loci suppressing and enhancing appetite, what did you find?
Right. So first of all, that fish, even larval zebrafish, regulate their feeding based on their hunger state. This wasn't clear. So people thought that maybe they eat reflexively whenever they can until they burst, that it's not under homeostatic control. Right? So the first thing we found is that when fish have eaten a lot, that they get satiated and then stop eating. And this particular center in the hypothalamus, in the caudal hypothalamus, that also mammals and humans have, was somewhat confusing, because in mammals, in mice, there were 2 conflicting theories about how, in particular, serotonin, which is one of the neuromodulators in the mammalian brain, how this correlates with hunger. So some studies found that if your hunger goes up, your serotonin levels go up. So this positively correlates. And other studies found the opposite, that if you get hungry, your serotonin levels go down. So there was some conflict. What we found in fish was a little bit more complicated, but very illuminating. What we found that in the caudal hypothalamus, so initially serotonin levels would go down when hunger goes up. So this would mean that serotonin codes for satiation.
Yeah.
So when fish gets hungry, serotonin levels go down. That's what we found, and serotonergic activity went down. Yes, sir. But then when we just exposed the animals to food without allowing them to consume it, right? So if they're just getting exposed, if they can smell and see the food, we found that serotonin levels instantly jumped to a very, very high level. Yes, sir. And then when the fish starts feeding, these serotonin levels were decreasing again back to baseline levels. We found both, right? In that case, when food was available and the fish could eat, then serotonin coded with satiation, yes, with hunger, right? So when hunger goes away, the serotonin levels go down. But if there's no food available, yes, then the serotonin level coded for satiety. So it depends, right? It told us that there are two ways of being hungry, namely being hungry in the presence of food or being hungry in the absence of food. And we can all relate to that, right? So even humans know that, right? If you're hungry and there's no food present, then being hungry is a bad state, internal state, and you need to explore and scavenge for food. Right? So this is really a stressful situation that probably also involves the sympathetic nervous system. So it needs to energize you, and you need to look. However, if you are hungry in the presence of food, you should not explore and run after and hunt and play. You should exploit, settle down and eat. Yeah. So you need to have a completely different drive when food is present. And so what we found is that what serotonin is doing, it either will energize you. Well, that's basically what we think, yes, that serotonin will energize and drive you to look for food, yes. But then when food is present, it will stop and it will allow you to exploit the food that's present and consume it. So this switch this homeostatic switch in the context of appetite and feeding, it was very interesting to find this even in larval zebrafish.
So my next question is, how does zebrafish use a graded stochasticity algorithm to predict and optimize their hunting strategy of paramecia and improve their noise-free strategies?
Yeah. So this is when you look at what they do when they feed. They don't. They hunt moving food, right? Like tigers, like predators.
Yeah.
So the unicellular organisms, the paramecia that they hunt, move themselves. So they actually need to chase them. And what we found is that they don't do a targeted approach. They use a somewhat stochastic approach, right? They sort of incorporate the fact that the prey is moving stochastically. So you cannot predict where the prey will be in the next instance. So what they do, they use sort of a biased approach where they slowly approach the angle. They don't go directly for the target, but they go halfway there and do it iteratively.
Okay.
Yes, sir.
So on a more broad scale, to what extent do you think artificial neural networks can give insight into biological neural networks?
Yeah. So how do artificial neural networks, what can they teach us about biological neural networks? I think a lot, Esther, because the parallels are uncanny. Yeah, I think the biggest progress is made by actually building the neural circuits that we think make up the brains by replicating or emulating them in a artificial neural circuit. Yes. And if we find that the resulting computations are very, very similar, then we are very close on the path of discovery. I think that is the biggest advantage. Like what Richard Feynman said, right? If you think you understand something, you have to be able to build it. I think this idea of rebuilding the circuits that we think we have understood in a biological brain in a artificial neural network gives you insight, but it also gives you confidence that you're on the right track. And it also will tell you, if it doesn't work at all, that you have more research to do.
And in this case, how did you use the accessibility of the artificial neural networks to discover a new temperature-responsive cell type in the zebrafish cerebellum?
Yeah, so I mean, that's what we did, right? We thought we understood how temperature sensing in the zebrafish brain works. And then we built we took the constraints of the biological zebrafish brain, we structured the artificial neural network according to these same boundary conditions. And by training this new constrained artificial neural network, we tried to reproduce the temperature-dependent behavior of a larval zebrafish, and that was successful. And then what we found is that by studying this artificial neural network, we found that it had generated, by training, a new cell type, a type of nodes in the artificial neural network that was necessary to generate the behavior, but that we hadn't found in the in the biological brain before. Yeah, but because it emerged in the artificial neural network, we could do a targeted search again in our biological brain, and then we found them. When we looked more closely, we actually found that these neurons also existed in the biological brain, and that was it just turns out that that's a useful tool, right? Then the artificial neural networks become a discovery tool for finding specific elements in the biological brains as well.
So you also have the tool of the random dot motion discrimination paradigm. And so how did you use the zebrafish's optomotor response as a measure of decision making?
Yeah, so this is a classic, a very old assay in human psychophysics, also in monkeys, where you show either people or monkeys random dots on a screen, where a small percentage of these randomly flickering dots move coherently in the same direction.
Right.
And the human observer or the monkey needs to decide whether they go right or left. That's the task. And if you do this in humans or in monkeys, you find that you can actually uncover a lot of things about the strategy we use, right? That we integrate information. So this is usually done at a level where it's difficult, right? Where only 2 or 3% of the dots move coherently. But humans can do it, but they need to stare at the pattern for a relatively long time before they can make a decision. And the insight you can get from that is that we are integrating information. By staring at it for a long time, we accumulate evidence until we reach a certain confidence level, and then we make a decision. So it was relatively clear that monkeys and humans do that, and people have tried for a long time to construct neural circuits that underlie this integration and thresholding process. And what we found in fish, that fish do exactly the same thing, but they do it reflexively. They don't, you don't have to tell them to report the direction the dots are moving. Fish will reflexively follow with whole field motion. So you don't need to train or teach them. They will do this automatically. So we could use this innate reflex, that's the optomotor reflex, to ask them about the underlying decision-making process in these random dot paradigms.
Well, thank you very much. That's all my questions, and your answers were absolutely fascinating. And the work
Good.
you're doing is really important, and it's going to reveal so much for future research for people of my generation. We're going to use your reports that you made in your papers, and we'll hopefully expand and everything. So thank you very much for joining us today. If you have anything else you'd like to talk about.
No, I think this is everything. I just wanted to also say thank you, Martha, because I think you are actually doing an equally great service to the world, because one thing that we are really bad at, scientists, is communicating our research to the broader public. Yeah? And for that, people like you are immensely valuable, right? I think we need people who take our research and process it for the general public such that they can also benefit. So let me also thank you for making this possible.
Thank you so much, and thank you everyone for listening.